Okay, everyone. This is going to be the first video. Most of this is going to be just kind of going over sort of how to prepare for this sort of class, as well as some of the tips and tricks to sort of get through a lot of the material. Not just get through it, but really internalize it and make sure that we're really internalizing and knowing this material. So first thing I'm going to recommend is that watch all of your lecture videos. I know it can be a little bit difficult, especially at first, when there's a lot going on. I highly recommend doing something like this where you have a schedule and you go through and you input all of your information that you need. Here, this is my setup for this is try 6. Where I go through, I have my videos, PowerPoints, what assessments I'm going to have that are repeating, add any additional assessments each week, and I'll include, for example, how much time I expect each thing to take, so that way I have a general idea sort of what to expect for that week. Okay, we're getting into the nitty-gritty of biochemistry now. The slides, especially in this specific class, the slides is going to have almost all of the material that you need, which is why one of the reasons why I recommend watching the lecture material first. Some teachers have accents. This goes for all classes, and that's fine. As you get through, at first it can be a little bit difficult to maybe understand their specific accent, but give it some time. And after 2 or 3 weeks or so, as long as you're keeping up with all of the material, you should get the hang of it, and it'll be a lot easier for you to understand and even be able to speed up some of the videos. So, all of this material, all of these slides, specifically with the biochemistry classes that taking here at Parker, the slides all come directly from his book. Now, you can, as well as saying, there's kind of 2 methods. You can watch, I would recommend watching the videos first, get some good ideas here, and then jump over to the book to really solidify the information. So for most of these videos, we're just going to take a look at the book and kind of reference the slides as we go through. But this is the book will be one of your best ways to learn and understand this material. And this is kind of how I would recommend reading any textbook. Dr. Sarkar specifically has set this book up really, really nicely and really simply, but the general ideas and concepts will definitely apply to any textbook that you're going to be reading from here on out. Okay, so biochemistry, first of all, the chemistry of life, the cell. The cell, we're going to be concerned with the properties of the biomolecules and the reactions of the biomolecules. Now, this is a very important thing to notice. As you start reading a textbook, vocabulary is going to be huge. This is with any of your slides, anything else. So anytime that you see a word, so for example, biomolecules, that's one of those things where I'm like, okay, what does that actually mean? I'm immediately going to go over to my dictionary, define it. A molecule that is produced by a living organism. Okay. Bam. Throw that in there so when you go back to review, it's right there. And as you continue to acquire more information, more vocab, everything's going to keep getting easier and easier. Okay, so we have properties of biomolecules. So molecules produced by living organisms, so we have the properties of those molecules. And then we have the reactions of those molecules, so how they're going to interact with each other. And just a nice little chart giving us a little bit of an overview of the cell. Talking about the nucleolus. Again, just vocab word, nucleolus versus nucleus. The ribosomes, peroxisome, all that sort of stuff. This is a good point where I would take a pause and take a minute, define a lot of these terms if you don't remember what they are, and make sure that you know what those terms are, even just so you have the definitions here so you can easily look back later and see it. Okay, so we have the characteristics of life. We have complexity, extraction, transformation, and systematic use of energy to create and maintain a functional life form. Break it down a little bit easier. Extraction, pulling out, transforming, and using energy to maintain a functional life form. So this is talking about our ATP system, and we'll get into that later on in the trimester. So ability to sense and respond to environmental changes. So think of some of the changes that we can have in our environment. If it gets hot, if it gets cold, if it gets wet, if it's dry, our body's going to change and going to adapt and respond to all of those different environments, as well as it can be an environmental change being like an injury or something like that as well. Capacity for defined self-replication while allowing change for evolution. So self-replication, obviously all of our cells are going to continue to self-replicate, but also allowing change for evolution. So if we're in consistently in, let's say, in high altitude, you'll get a lot more into this when you get into the classes of physiology, but our body is going to adapt to the conditions that we are in. So high altitude, for example, we're going to be able to regulate especially our red blood cells. We're going to get more red blood cell production. We're going to have an increase in oxygen carrying capacity. These are going to be the evolution, the adaptations our body's going to make in order to help us survive the best. So complex life. Living organisms have these two things: special structures with distinct functions. Like I've said, looked at all of these up here. Huge number of different compounds from simple to complex. We'll get into some of those. And molecules capable of highly specific complex interactions. So those biomolecules, that's what we're going to be talking about, the reactions of those. So before we continue, we want to look at some of the fundamental concepts of chemistry. Hopefully these are all things that you have a lot of definitions and sort of stuff that you've learned previously, and this is just going to be a quick review over them. So elements, atoms, and molecules. Elements, first of all, we have our table of all of our elements that we're going to have. So these are going to exist in their individual natural forms, as well as later on they'll also be involved in our compounds when they're together. So atoms are the smallest known unit of an element, primarily composed of electrons, which are negatively charged, protons, which are positively charged, and neutrons, which are neutral. Exactly, we have electrons negatively charged, protons positively, neutrons electrically neutral. So the molecule, so we have atoms, a molecule, joining of elements to form a stable structure. So for example, if we have carbon dioxide, that is now a molecule because we have a couple of different elements together to form a stable structure, or water, H2O, all of those good stuff. So inorganic versus organic molecules. Again, here, I don't know the exact definition of this right now. So define inorganic molecule. You're going to love, love, love Google. So inorganic molecule is a molecule not consisting of carbon atoms. And then if we look at organic molecule, are molecules that are made of carbon and hydrogen and can include other elements. This is the biggest thing, is the better you can prep your material, easier to go back through, the easier it's going to be to study, to review. So the inorganic molecule is the molecule not consisting of carbon atoms. So anything that's no carbons. Organic molecules have the carbon and the hydrogen is kind of as their base, and they can include any other additional element. Okay, now we have our atoms. Atoms, we have the nucleus with the protons and neutrons, and then electrons around the outside. And we have our valence rings, as we can see here. Typically, what we want to see is that the number of protons is going to be equal to the number of electrons. That's what we want to have a balanced molecule. Now, it isn't always going to work out that way just because of the way that electrons and these rings called the valence shells or orbitals are going to want to be. They want to be in the most stable state possible. So sometimes they're going to get rid of electrons, sometimes they're going to get extra electrons, making, in general, certain atoms positive or negative in nature. So electron shells or orbitals are layers around the nucleus. The number of shells depends on atomic number. So the atomic number, I think that is the number of protons. And again, like I can say that, always 1000% good to double check. Atomic number, fine. There were protons in the nucleus of an atom, perfect. Okay. The first shell, so we're gonna have different multiple shells. So see how there's one shell here around the hydrogen, 2 here around the carbon, and then we're going to have even more shells out that hold more and more of these electrons. And each of them is going to hold a specific number of electrons in it. So the first shell can only contain 2, the second shell can contain up to 8 electrons. So that means we get, when we have 2 shells, we have a maximum of 10 electrons. So if we go back and we look up here, 10, right there, neon is going to be our max. So all the way from hydrogen to neon, we're going to be filling up our first and second shells. Okay. Electrons in more distant shells have higher energy. This is going to be important later on, knowing the ones in the outside have higher energy. Valence electrons, you might heard me mention valence shells a couple of times. So valence electrons are those in the outermost shell. And these are the ones that participate in the chemical reactions and form bonds. So these are the ones that we really actually care about when we talk about biochemistry and when we talk about reactions, are these valence electrons. So we have a couple of different types of chemical bonds. We have electrovalent bond, covalent bonds, which are broken down further into nonpolar and polar, as well as hydrogen bonds. So these are all going to be key. Remembering, it's electrovalent. Another name for it is ionic. And we'll get into a little bit of what each of these individually mean. But right now, it would be a great point. If you don't remember what any of these mean, might as well stop. Even though it might tell us later, a little bit later on, its repetition is key. So if we want a specific definition for electrovalent bond, define. Ionic bond, also called electrovalent bond, is a type of linkage formed from the electrostatic attraction between oppositely charged ions. So attraction between oppositely charged ions in a chemical compound. Boom. Covalent bond. And you're going to see that this is going to be really repetitive doing this for a while, but trust me, it works. It helps so much. Covalent bond consists of the mutual sharing of one or more pairs of electrons between 2 atoms. And we get into nonpolar versus polar. You can look at those up on your own, as well as hydrogen bonds. Hydrogen bonds, a little self-explanatory. It is between hydrogen atoms. Okay. So talking about the electrovalent bond or the ionic bond, the valence electrons are transferred from one atom to another, forming charged atoms, also called ions. Any atom that loses electrons becomes a cation or positively charged. We know these definitions. Cation versus anion. This will help you so much later on down the road, and knowing that which one is positive and which one is negatively charged. So for example here, our sodium atom is going to donate, because it only has one in its outermost shell, and that's not very stable. It wants to have a full and complete shell. So naturally it's going to want to donate that electron to something like chlorine, so that way it has it in its outermost shell, so that way it can complete its outermost shell. And by doing this, that's actually going to leave sodium with a net +1 charge because it's losing that electron. And then the chlorine is going to have a -1 charge. And like we say, opposites attract, so we will actually get a bond formation between these two because the positive and the negative will attract to each other. Okay? Just like we said, the bonds are formed by attraction of the positive and negative charges, and the ionic bonds are weaker than covalent bonds. So we're going to get into a little bit of a tier list, kind of what bonds are stronger versus which ones are weaker. The ionic bonds are weaker than covalent bonds. Throw that in the back of your head, write that down, star that, highlight that, because that will keep coming back in a lot of other questions that we start to ask. This is dissociates when dissolved in H2O. So if you take a compound like sodium chloride, for example, and you put it into water, the water is actually going to dissolve it and break those bonds. Covalent bond, when atoms share the valence electrons, and here we get into polar versus nonpolar versus polar, right? Nonpolar is when the covalent bonds equally share the electrons. So this is going to happen typically with two atoms that are the same. So you have like hydrogen and hydrogen, or oxygen and oxygen, where they have the same what's called electromagnetic strength. So if you look up a periodic table of electronegativity, let's see if we can find a good one. Here we go. As we move along our periodic table, we have increasing electronegativity from the left to the right. So as we go more and more and more to the right, we have higher or greater electronegativity. And as we go from the bottom up, we have increasing electronegativity, meaning that these ones up here are going to have a stronger pull on those electrons. So for example, oxygen right here, oxygen we usually typically consider pretty highly electronegative, that it really wants to share, it really wants the electron. So if it's sharing with something besides another oxygen, it's really going to pull it forcefully towards it and hold it more than anything else. So there we go. Hydrogen, so non-polar covalent, meaning that both of them have the same pull on the electrons. And polar bonds means that they share electrons unequally. So something like water, oxygen is way more electronegative than the hydrogens. So the electrons are going to be pulled much closer to the oxygen. Coordinate covalent, a bond is formed when one atom provides both electrons in a shared pair. This is just more going into the definitions. A bond is formed when one atom provides both electrons in a shared pair. So if one of the atoms is providing both of the electrons, for example, for the hydrogen, so this hydrogen, so the nitrogen has two spare atoms right here, and it's going to hold both of them, so the hydrogen comes in, doesn't contribute any electrons, and binds in. That's what the coordinate covalent is talking about. Hydrogen bond, the hydrogen atom forms polar bonds. It's important to remember that. Polar bond with another atom, it takes on a slight positive charge. A lot of these things are a little tedious, like memorization type things, which they're really good to review. I would recommend putting out on a slide, like taking all these little tidbits of information, putting those to the side, kind of in a little review chart to go over a couple of times. Okay. So hydrogen atom forms a polar bond to make it attracted to any nearby electronegatively charged atoms, and these are called hydrogen bonds. So they form between adjacent oxygens. This is the one that we'll mostly see, that oxygen. So these hydrogens that are now slightly positively charged because the electrons get pulled more closer to the oxygen are going to bind. The hydrogens of one molecule are going to slightly bind to the other oxygens as well. And this is why we use water a lot. Water is kind of special. Water is really, really cool. It's right. A lot of our body is made up of water, so we have a lot of these different hydrogen bonds at work in our body. So water's special properties. It's obtained due to its polarity. We talked about how the oxygen is really going to pull all of that, the electrons towards it, make the hydrogen more positive, create those hydrogen bonds. And because of that, it has the hydrogen bonding capabilities. Water is a solvent. This is probably one of the biggest things to remember. Solute versus solvent, right? The solvent is what we put the solute in to dissolve it. The solute is the compound or substance that we are putting into the solvent, typically water. Super easy to get this mixed up. Know these definitions. This is another one. Really make sure you know those like the back of your hand, so that way you don't get those confused or mixed up later on. Okay, now we have a couple of more terms: hydrophilic versus phobic compounds. Hydrophilic, so this is what like dissolves like. So hydrophilic, the root philic. So hydro is water. Philic equals like to like. What actually is the? We should get the proper definition. Denoting a fondness for a specific thing, so liking it, and a fondness or an affinity. And then phobic, like a phobia, is to fear, not like. So something that is hydrophilic, means it likes water, is going to, they are soluble. Soluble means that they will dissolve, that they'll break down. If you put it into water, it breaks down. Hydrophobic compounds are insoluble in water, which means that, like oil is hydrophobic. So oil or fats. So if you put oil, or if you've ever mixed oil and water, you'll notice that they completely separate. That the oil will stay on the top and the water's underneath. If you've never seen this, go and just put some oil in water and watch it separate. Really important to remember these, as well as we'll start to get into different compounds, and we need to remember what does it mean to be soluble versus insoluble, hydrophilic, hydrophobic. Okay, effective extracellular solutes and water movement across a plasma membrane. Lots of words there. So extracellular solutes. Solutes, we had talked about, it's the compound or substance that we're putting into the solvent, into the water. Extracellular is just going to mean outside of the cell. So we have in our body what's called interstitial space. Where in between we have all of our cells, and then outside of our cells we have intracellular fluid, and then outside all of this blue stuff here is our extracellular space. So it's outside of the cells. On water movement across a plasma membrane. So plasma membrane, you'll also see this called a semi-permeable plasma membrane. So the plasma membrane is what makes up the membranes of our cells, the outside of our cells. And we'll see these type of pictures a lot later on, where they're phospholipid bilayer. They have a phospholipid bilayer. And these are semi-permeable. So we'll get more and more into that, but just if this is something that you don't quite remember, jump to YouTube. Again, one of my favorite things to do, and literally just look up phospholipid bilayer simple. And look, couple of 2-3 minute videos, cell membrane structure and function. Watch a little quick video on it. And I guarantee you that'll help you out. Like I said, the biggest thing is don't move on until you understand the material that you've gone over up to that point. Okay. Osmosis. So osmosis, whenever you see this word osmosis, you're always talking about water. You're talking about the movement of water specifically. Always the water is always what is moving. So it's a spontaneous movement of the solvent, or what we talked about up here. Solvent is what we put the solute in. The solvent is the water across a semi-permeable membrane. Semi-permeable, if you don't know what that means, quick definition, that means it is partially able to be passed through. So certain things and some things are able to get past it, but other things cannot. Okay, so water crosses a membrane into an area of higher solute concentration. So remember what solute is? I'm trying to repeat the definition. The solute is the stuff that we dissolve into it. So the sodium chloride. So if there's a higher concentration of it on the other side, meaning that there's more of those particles there, it's going to go. The water is going to travel to the area of higher concentration. Eventually, the tendency to establish an equilibrium of concentration on both sides of the membrane. So what this is saying is that we have a semi-permeable membrane right here, right? We've got stuff in both of these, but one side has more stuff. So our water is going to travel through that semi-permeable membrane to the other side, so that way they're have equal concentration. So the water to stuff ratio is going to be equal to each other on both sides, on either side of the membrane. Okay? So the force for this movement of water is driven by differences in osmotic pressure across the semi-permeable membrane. We'll get an osmotic pressure in a minute. And plasma membranes are more permeable to water than compared to other solutes, such as ions and other macromolecules. Macromolecules, we're talking about things like our carbohydrates, like glucose, our proteins, as well as our fats, our lipids. And we'll get into those. So jumping down to the osmotic pressure. Osmotic pressure is described as the pressure required to maintain an equilibrium across the semi-permeable membrane with no net movement of solvent. Try and break this down to make it a little bit more concise, a little more clear in your own words. So osmotic pressure is pressure to keep balance across membrane. So no, remember, solvent, water, moves. So there's a pressure, kind of like you think about whenever there's water, like even like a container, the water is pushing against the container is pushing back. So that's sort of that same similar idea where there's a pressure, the water is pushing, but then there's a pressure on the outside keeping the same amount of water in. Okay? Osmolarity, describing the solute concentration. Solute concentration, that's the concentration of the stuff, which can exert a specific osmotic pressure on the cellular membrane. So the more stuff that we have, more stuff equals higher pressure. And think of it like that, if there's more stuff, it's pulling the water towards it. So if there's more stuff on one side, it's going to pull the water towards it. It's going to create more pressure to pull that water. Isotonic solution. If a solution that contains the same osmolarity, like the cytosol of a cell, When an isotonic solution, iso meaning same, tonic it's typically using for tone. A solution is just a mixture of a solvent and a solute. So it is a solution that contains the same osmolarity like the cytosol of a cell. So it's balancing like a cell. When an isolated cell is present in an isotonic solution, the cell does not shrink or swell. So basically what that's saying, remember we talked about intra versus extracellular? So you have a solution in here, and the concentration of the ratio between solute and solvent, so stuff to the water, on the outside, let's say on the outside it's like a 2 to 3 ratio. On the inside, we have the same ratio of stuff to water, meaning that there's not going to be any pressure pushing water in or pulling water out. Okay? So nothing's going to happen. It's not going to shrink or swell. Hypotonic, the way that I always remember this, I think hypo, I think hippo. When you put something in a hypotonic solution, so it is a solution in which the osmolarity, so the amount of stuff outside the cell, is lower than the cytosol of a cell. So water concentration is higher than the solutes. When a cell is put in a hypotonic solution, the water molecules enter the cell and the cell swells. So hippo. When you put a cell in there, it blows up because the water is going to be coming in because there is more stuff inside the cell than outside, and it tries to balance, so water goes in. Hypertonic means it has more on the outside. So when the cell is put into hypertonic solution, the water from the cell moves out, and as a result, the cell shrinks. And you can see examples all down here. Yep. Cell in hypertonic solution, the hippo, big and puffy. A little typo here, so make sure that's hypertonic solution. Okay. Continuing on with more reviews, acids, bases, pH, and buffers. A lot of people tend to struggle with this sort of stuff. So we'll use very specific definitions for acids and bases, and we want to make sure that we keep those solid because there are actually different definitions for acids and bases depending on what what textbook you're referencing. So for our purpose, we're going to look at this way that acids are compounds. So compounds, take a minute to think what compound is. So that is different atoms together, right? That releases protons. That's the big idea right here. Releases protons in a solution. So it is a proton donor. Later on, when we get into pH, when you increase the amount of hydrogens, you actually decrease pH. Okay? We'll get to that later, though. So a proton donor. Remember that. Examples like hydrochloric acid, sulfuric acid, phosphoric acid, and carbonic acid. Bases are our proton acceptors, or anything that lowers the hydrogen levels of a solution or releases hydroxyl ions, are OH minus. So, because when you release an OH minus, just like that, if there are hydrogens around, remember our negatives and our positives are going to attract to each other, and then we'll end up with H2O, which neutralizes our hydrogen. So it's going to increase our pH. Okay? Because we'll look for that word, that hydroxide is a good indicator. Acid versus hydroxide. Ionization of water. So this is water can go through this process where it can go from H2O, breaking up into a hydrogen and a hydroxyl, or a hydroxide. And it can go back and forth between those two. So ionization of water is quantified by ion product of water, Kw. This is going to be a constant. And the basis of the pH scale. H plus concentration in an aqueous solution. Now, remember, aqueous, I wanted the good definition for aqueous, because it's our first time seeing that word. Aqueous, of or containing water, typically as a solvent or medium. Bam. So it's a solution with water. Aqueous solution. Okay. We have Kw equals these little brackets. When you see brackets like this, that is referring to, I'll just put an X in there as a placeholder, concentration of X ion or atom. Look at the concentration of that. Those little brackets mean concentration of whatever's inside. Okay, so pH describes the hydrogen concentration of a solution, so it tells us how much hydrogen that there is. It's from 0 to 14. A lot of this is just kind of some math stuff. We won't really get into a lot of the math and like the calculations. We don't really care too much about the actual calculations of it. We'll do some calculations, but nothing super complex. It's more just as long as you can understand the concepts behind it, you can just plug in the numbers and you're fine. So acids have a pH lower than 7, and bases have a pH greater than 7. Very important to remember that. Where if we put an acid in a solution, it lowers our pH, and bases will increase our pH. The normal pH range of blood is between 7.35 and 7.45. This number will keep popping up in your other courses. Really important to remember that number, as anything above or below this essentially means death for the human body, which our body is amazing and is able to regulate this really, really well. Okay, so talk about acids, talk about our bases. Now buffers. So buffers are what we're using to adjust those acids and bases to get them to where we want to be. So pH changes a little when a small amount of strong acids, when we talk about strong versus weak acids, so strong, meaning that strong means that it completely dissolves in the solvent. So something like hydrochloric acid, for example, is a strong acid because it completely dissolves, all of its hydrogen dissolves. You'll also see some things being described as weak acids, and that means it partially dissolves. So if you put or even just weak acid or base, partially dissolves. So if you think about putting an acid, so if we put a strong acid into water, pH goes down by a lot. Let's say we put 10 milliliters of strong acid into water, pH is going to really drop down by a lot. If we put 10 milliliters of weak acid into water, the pH goes down a little bit. That's just the describing that definition, knowing the difference between strong and weak. Same thing for if we had bases. A strong base, pH goes up by a lot, and a weak base, pH goes down, goes up by a little bit. Okay. So the definition of a buffer, it's composed of a weak acid. So remember definition over here, weak acid, and its conjugate base. Or a weak base and its conjugate acid. So our conjugate base. So a conjugate base contains one less hydrogen atom and one more negative charge than the acid that formed it. This is talking about the buffer. When you put in a weak acid, when you put the weak acid into water. Same thing with conjugate acid or conjugate base. When you put that weak acid into water, so that weak acid, H2CO3, it's going to break down into its hydrogen and then its conjugate base right here. Meaning that this base, remember we talked about base, what can a base do? A base is going to accept hydrogen. So our conjugate base is formed from our weak acid, and it's going to be able to if it needs to in that situation accept hydrogen. Same thing with the weak base and its conjugate. As we put the base in, it's going to give off the OH, and then it's going to be left with a conjugate acid. So we have a weak acid. You have the conjugate base of that with the hydrogen. So for example, acetic acid, and then acetate is its conjugate base. Some of these we'll give some examples of these in like the book and in the PowerPoints. So the ones that are like the most common, especially the ones that Dr. Sarkar mentions, just look at those specifically. You don't have to memorize a ton of them. If you know what the acids are and you can identify, okay, is this a weak acid or a strong acid, you'll be able to then understand, like, okay, what would its base be? Because acetic, acetate, pretty similar spelling. It's kind of the way it's going to go. So for weak acid, dissociation constant is determined by the expression dissociation constant. So this is a constant. This is something that's been tested and discovered. Each of their different compounds are going to have a dissociation constant, which you need like a little chart, and it'll give you all of like the chemical compounds and then what their constant is in order to do these sorts of equations. So we won't worry too much about that. So here we're talking about that Henderson-Hasselbalch equation. I just know the equation in general right here, where the concentration of hydrogen, conjugation of the conjugate acid over the compound itself is going to give us that dissociation constant. And then all of our other extra maths and such to give us the pH equals pKa. So, again, this is something else that I would, if it's really confusing and you really want to know all the ins and outs of it, just look up a short YouTube video on it, and it will help you get what you need for this course and for, honestly, for most of what you're doing. We're not going to be biochemists. So, some of the important buffer systems that we will eventually look at: the hemoglobin, the bicarbonate, phosphate, and the protein buffer. Carbon. Biomolecules are carbon-based. We are organic life forms, so everything we're going to be made up of is going to be made up of, it's going to be containing carbon. Some other important elements: hydrogen, oxygen, nitrogen, phosphorus, and sulfur, as well as metal ions. Let's see, if you take a multivitamin, all of these you'll see in the vitamin that we need for these. And there are more than 30 elements essential for life. Carbon, whenever you see a structure drawn like this with just like the sticks off of it, if nothing else is shown next to it, what this is representing is that there's a hydrogen bonded to each of these spots. Okay, so these are the lines that represent a bond, and hydrogen, if it's left blank, it just means that there's a hydrogen there at the end. Versatility of carbon bonding. Carbon will bond to a whole bunch of stuff, really good for bonding. Double bonds, single bonds, all of that good stuff. So we have functional groups. We're going to have our biochemical reactions involve the functional groups of molecules. So when you see different molecules, these Methyl, ethyl, and phenyl, you'll see these are prefixes, and then there'll be something else afterwards. All that's saying is that, so R represents more stuff. We don't necessarily know exactly what it is unless we have the full compound. So R could be another C, 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 O, O, C, H, H, H. What we know, so this right here is our R group, and this is our functional group. So any of the reactions are gonna take place over here on this side. This is where the reaction is going to occur, and where we really want to give our focus. So you can have methyl, ethyl, and phenyl are just three of the really common ones, and we'll see more as well. So all of these are functional groups. All of these are functional groups that we'll see as we go through and see, especially when we get later on into like amino acids, we'll look at a lot of these functional groups and how they interact with each other. See, amino group, all of these are going to slowly start to pop up. You don't need to memorize these like exactly for what they are. I would take a minute and observe them and look at them. Maybe practice writing some of them down just so you get a feel roughly for how these things are written. But don't worry too much about memorizing every single one of these, because in reality you're never going to see these in real life. It's just understanding kind of what they have in that functional group. So carboxylate has the carbon and the 2 oxygens, meaning that it has opportunities to bind with things that would bind with oxygen. So it's just kind of looking at it like that. And also carboxylate, if you look at it, CO2, CO2, pretty important and something that we'll see a lot of later on down the road. So we'll see carboxylate reactions later on involving the release of CO2. Functional groups in a single molecule. This is just showing you we can have molecules can be huge. So we have all of these different groups here in this one single molecule, acetyl coenzyme A, acetyl CoA, which plays a very important role later on in the glycolytic pathway and the Krebs cycle that we'll get into. And that is the full chapter 1. So like I said, up here a little bit more as well in this book, there are these questions right here. Some good review, just a workbook. Introduction to Biochemistry and Review of Basic Chemistry Concepts. Define atom. I would go through these as much as you can. If you can't put these into your own words, that's the most important thing, is putting things into your own words. So if you cannot define this in your own words, you need to find a definition and put it into your own words. So let's say define atom. So an atom is the basic unit of a chemical element, source of nuclear energy, an extremely small amount of thing or quality. So the basic unit of a chemical element. Is that like the best one? Maybe not. I like this more. The smallest part of a substance that cannot be broken down chemically. Each atom has a nucleus made up of protons, positive particles, and neutrons, and electrons move around the nucleus. I love that definition, and I wouldn't tweak it at all because I feel like it fits well with me. But even the next question, the fundamental subatomic particles present in an atom are, we have those protons, neutrons, electrons. The electrical charges present in the above-mentioned subatomic particles are, remember, protons pro-positive, neutrons are neutral, and electrons are negative. What is a cation? How cations are formed? How are cations formed from an atom? So these kind of questions, going through these questions and answering them, especially if you can do this before that first week of school, very, very helpful. I'd highly recommend doing this. So yeah, that is this is going to be our first part in the series. Hopefully I can keep going through as much of the information as I can. I'm going to try and get through a decent chunk. But just the way that I just did this right now, this is how you can go through these reading materials on your own. So it took about 50 minutes. Hopefully some of the chapters will be longer, some will be shorter. And as you get quicker on this, you're going to have to look up less words, less stuff, less definitions. And really when you come back to review this, look at all these beautiful notes that you have right here that you can refresh yourself as you go along. So study hard, and I wish you the best of luck.